Today we'll be seeing how this little tiny coliphages can be used as proxies or the sentinels to map the viral contamination in the drinking water and how Nanopore helps us to achieve this. Thank you for introducing me, but this is like a brief background, how I got here exactly. So basically, my PhD dealt with the Ganges River, and at that point of time, I used Nanopore to deal with the metagenome, microbiome, virome, and the phageome of this riverine system. And this is how I gained expertise in that, and now I'm using those expertise to translate the ecological studies into the next generation drinking water security applications. And here in Cranfield, I'm working on the drinking water treatment plants and assessing how this virome studies can be useful to tackle the viral contamination. So this brings us to one of our key questions, that is, okay, what we choose to measure, it determines what we value, and what we value will shape our actions. So this is a quote that I really like, and I've taken it from Stiglitz and Fitosi. So until now, our challenge was that bacterial indicators are what we chose to measure, the bacterial contamination. But the viral contamination is something that the landscape was ignored, or it was not being tackled by the bacteria. And therefore, now we are choosing another somatic coliphage as a proxy to measure this type of contamination. So I would like to start my presentation with this code. So the standard for a safe drinking water for the Escherichia coli, the coliform count should be like a 0 colony forming unit per 100 ml. And similarly, for the somatic coliphage, now it is like a 0 plaque forming unit per 100 ml is the standard that is given by European Union directive, and it is also backed by the ISO standards. So this is what you can see, like this is a picturesque form which I wanted to present of the directives that have been focusing on these two microbes. So yes, so basically now how these plaques look like, that is how they look like under a microscope, but this is how they look like when it is visible to you on a plate. So these tiny dots which you can see, these are actually these phages that are present over there. So they are very tiny, but they tell you a lot of story. So first is why, because they are globally relevant now. They are being recognized increasingly by European Union's drinking water directives, and also by the International Organization of Standards. The thresholds have been defined that the coliphages have to be like 50 plaque forming unit in 100 ml of the raw water that you're being using to treat through the different interstages, and for the final water it should be 0 plaque forming unit. They are safe and specific. Now when I say these can be used as viral proxies, so it would be like viruses can harm you, but these phages, they are very specific to bacteria and they do not harm humans. And that's why they are safe and specific to use. They are better than the bacteria alone. Like I said, like both of them, when they are in a same team, they can help you for effective drinking water monitoring. They are the smart sentinels, and also they align with the Sustainable Developmental Goal 6, that is clean water. So the gap, that is, we don't know this diversity of the somatic coliphages because they have been newly introduced. So how they behave during the treatment is what we are seeing. So to answer that question, we have focused on 13 different drinking water treatment plants in the UK, and we have sampled the source water, like the raw water which enters into that drinking water treatment plant, the reservoir water, the different interstages, that whichever the drinking water treatments are being used, and up to the final water. And we were also interested, and we have explored the backwash water as well, in order to see which different types of the coliphages are being trapped and how do they change with the different treatments. So this is a snapshot of what we did. So right from the raw water till the reservoir and different interstages, we collected from 10 liters to 600 liters of water. We concentrated that water and took like up to 7 to 10 ml of the water was remaining after the concentration. That concentrate was then we had used a double agar overlay method in order to see the plaques that develop on this thing, and then we had individually picked up a single plaque from the plate, and that single plaque was sequenced using MinION's nanopore sequencing. Now, we did not isolate this plaque because we wanted to see, when we put the water on the Escherichia coli and we get those plaques, what is the genomic diversity present within that plaque. Because these are the coliphages that are present in the drinking water. But what is the diversity of the coliphages? Now, the question for this is because we want to find those signatures. Some signatures that can be used, which are common between these coliphages, which can be used in further for real-time monitoring. So that was the whole aim. This will help us to achieve a virome-informed drinking water safety. So what we found out from this is like how many phages are present. So we can see in the raw water, the diversity of the phages are rich. And over the time, what happens as it passes through different stages and to the final water, the diversity of the phages decreases. So the raw and the early stage waters, basically, they have a rich phage diversity. However, the diversity reduces when it passes through the subsequent treatment stage. And this means that the phage diversity declines stage by stage. And it is not random. It is because of the ecological filter that happens through just different stages. Now the question is how these communities differ across the treatment stages. And over here we can see that each dot is a plaque which represents like the multiple viruses which we got through. And the plaques from the same treatment stage, they cluster together, and as the treatment differs, so you can see each treatment has its own impact and will affect different viruses, restructuring the viral community. And therefore, we see that specific survivors are there. So these are like, they will give you an indication of which phages survive which different treatment. And those phages can be used further for monitoring. So on the basis of this, what we have understood is that we have few signature viruses like the alpha-3 virus, Syncytia virus, and Gemsa virus. So these phages, which are common all over the sites, they can be used further as the indicator species. And of course, I'm showing a relative abundance data, but I have also done an indicator species analysis, and we're finding the similar trends over there. So this shows us that it reshapes who dominates in this like particular stage and who is staying and which persists. So the further analysis towards this was looking into a differential abundance, that which phage is actually staying and which phages they disappear after the treatment. So the side sex, it shows like it shows that it has a GSC and ozonation based, which is a really good treatment train which can you can use for because you can see most of the viral removal as compared to that of the other treatment stages. So this also tells us that which particular treatment is also more effective towards the removal of the phages. And therefore, it shows us which phage will be dominated and which will be filtered out. And on the basis of this information, what we got was like 23,000 or nearly 24,000 assembled genomes from 59 different samples. Of this, 5,000 genomes were used further because those were like complete circular genomes which we got out of the single plaque. Then 319 were high quality genome, draft genomes that we got from that. And of this, 318 were lytic phages, and one interesting was this lysogenic phage. And of that, so 5,157 genomes which we have got, all of them, they are lytic phages. And it is interesting to see because this 5X174, which is used as a sentinel of this somatic coliphage, it also has a circular genome. So it is interesting to see in comparison with this reference how this phage diversity varies and what signature we can get in order to develop a biomarker for the surveillance of drinking water quality. So where do we go from here now? So we have the rich and stage-specific phages that we have got, that we have the biomarkers, we have the dominant phages, which stays in the treatment, which is shifting, and thousands of complete lytic genomes, which is just going to give us a virome survey. It's like a launch pad that is we are getting for a phage-informed water security. I'm sorry, this animation is too fast, but it was not looking that fast in my presentation. So yeah, it's not just easy to like deal this all work alone. I have a great team who has helped me. Two of my colleagues are also present here, Ruben and Erin. So yeah, I would like to thank my university, Cranfield University, the Water Science Institute. That's Francis, Pete, Bruce, Suniti, Muna, who has all been associated to this project. Of course, Oxford Nanopore for giving me the resolution to look at the phages in the single plaque level, which is like phenomenal. So, and London Calling for giving me the platform to present my work. So thank you. Thank you so much.